Okay, greetings everyone. This video I will be explaining about CRISPR Cas9, so how it can be used for gene editing purposes and all those things you will be seeing in this short video. Okay, I am Arnab Salam, so I am undergraduate student at VIT University. So let's begin in the video. Okay, so CRISPR-Cas9, CRISPR stands for clustered regularly interspaced short palindromic repeats. Okay, so what all the essential components and all those things and all those stuff we are going to see in this video. Okay, so and one more thing, as you all know, yeah, CRISPR, the inventors of, that is the discoverers of CRISPR, Emmanuel Carpentier and Jennifer Doud, has been awarded with the Nobel Prize for Chemistry in 2020, and it's one of the recently what upcoming fields for gene editing purposes. Okay, so that's all about the introduction. Okay, so what is CRISPR? Basically, CRISPR is just an adaptive immune system of bacteria. So if you are aware of the immune system, so how you fight an infection, if you come in contact with a pathogen, your body recognizes it, and there is undergo a lot of immunological pathways involved, and finally you will your body will produce something called the mature B cell. So B cell, and after that the plasma cell is produced, which produce antibodies, and the memory cell is created. So which on subsequent what subsequent in exposure to the same pathogen, it will again what flood you with the antibodies. So that will antibodies will protect you from the pathogen. So this is almost the same. CRISPR is also the same, but it acts in the bacteria. That is a single-celled organism. Okay, so this is how CRISPR works. At first, this virus infects bacteria that is not a single-cell organism. That is called nothing but a transfection. The process is called transfection. So this genome, on first exposure, gets integrated with the bacterial gene, and forms the genome. So this genome is formed. Okay, on subsequent exposure, that is in this case, so the bacteria already has the genome, that is already has the viral gene embedded in it, right? So if on subsequent exposure the virus injects the bacteria with the that is its genetic material. So what happens is the CRISPR system just produces a double stranded break. So how it is going to make using all those information, biologically embedded information, is what we're going to see in this video. Okay, so and finally as the end of this double stranded break, the cell survives. That's the final endpoint. Okay, so basically CRISPR has three types, and the three types has three different types of molecules involved in it. So three different types of molecules involved. Yeah, but most vastly studied type is what that this type 2 one, type 2. So as far as my literature concerned, I could only find three types. So if you could find more, or if there is any update, just leave it in a chat box so that others can refer. Okay, so yeah, three types, and basically we are going to concentrate on this type 2, and the other two also, and each and every type has its own significance. Okay, but the type 2 is the most vastly studied one. Okay, yes. CRISPR system, what all it requires? So protein component is nothing but the cas protein cas protein, and the RNA component is the guide RNA. Target molecule is the matching genome sequence to which the guide RNA binds. Okay, so not whole of the guide RNA will bind to the target sequence. The target sequence, okay, and from where the guide RNA didn't bind, starts the PAM sequence. Okay, the next to the that is next, right next to that what target DNA, the PAM sequence, that is a series of nothing but a series of nucleotides, and it has its own significance. I will be telling in the video later. Okay, so PAM stands for protospacer adjacent motif, and all those things again I will be explaining in the video next. Okay, so CRISPR locus. What all necessarily the CRISPR locus or a CRISPR gene should consist of? CRISPR should consist of Cas operon. These Cas operons would code for Cas protein, and a repeat spacer array that would consist of the foreign DNA material that it has already gained from the what virus. So again, this tracer RNA is also a some part of it. So what all its importance? Yeah, we will be seeing. Okay, tracer RNA. First, I would like to wait. Yes, this repeat spacer RNA gives rise to that is repeat spacer array region give rise to RNA type of RNA called a PCR RNA or immature cRNA we call. Okay, immature CRISPR RNA. So this trigger region tracker RNA will code for this tracer RNA. So these both will combine and it is involved basically. Tracer RNA is involved basically in the processing of what CRISPR RNA to guide RNA. Let's say for simplicity. Okay, so and again this guide RNA is formed guide RNA and after that when the combines with the Cas protein, it makes the double stranded break. So that is the key point of our interest. Okay, yes, so now we're looking into the components of the Cas9 system. It has a gray region called RUVC region that cleaves the non-target DNA strand and Hutchinus, that is the cyan color. That is the target strand. That is basically this gray region will separate the non-target strand and the target strand. Okay, so orange region, orange. That is the PAM. This is the PAM interacting domain. Okay, so it will detect the PAM, and also the PAM varies from species to species. So it has its own significance. So this is another graphical that is representation of the same. This is the target DNA sequence. The guide RNA is bound to it, which is given in green. Okay, so and here the other sequence here is given in that is the complementary strand is given here, and the interactions are given. Okay, so this is how it works. Hmm. Okay, basically this HNH and RUVC is involved in the nicking activity. Nicking activity is nothing but the separation of the DNA, so that the guide RNA can bind and the Cas protein is have thus facilitates that. Okay, this is a different PAM sequence. Okay, PAM sequence has its own significance. Say for example, if the Cas protein and all those factors necessary to what facilitate a double standard break is formed, or there are factors. Okay, so and if there is no PAM sequence right next to it, so the double standard break will not be made. Say for example, here in pyogenes, the PAM sequence is NGG. So N stands for nucleotide, any nucleotide of choice. GG, guanine. Okay, so if the right next to the target site, if the PAM sequence is not encoded as NGG, the double standard break is not made in case of pyogenes. Okay, so likewise is the it is the concept applies to all the same. So that's the thing, and the if the PAM sequence, that is proto spacer adjacent motif, is there, then the double standard break is made between the target in between the target site. That is some 10 nucleotide base pairs within the that before the PAM sequence, not more than that. Okay, though it varies from species to species, as I said earlier. Okay, next. Okay, double standard break is made. So what all? So basically, in order to in mammalian cells and all those recent cells, that not the recent cells, sorry, all those evolved cells, eukaryotic cells, has its own DNA repair mechanism, so that in case of if there is any damage, also they can cope up with that. So such DNA repair mechanisms or non-homologous enjoining and homology directed repair. Okay, non-homologous. First, I will say about homology directed repair. So homology directed repair is nothing but double stranded. These are the two broken strands. Okay, and after that you are inserting something with some gene or some nucleotide sequence that is homologous to the broken pairs, so that these will go and bind up with this will go up and bind up with this. So this is homology directed repair, as the name insists. So this can be what used for repair, novel function, or therapy. So therapy, as you see, there might be some defects in the gene. Okay, so you remove that part of the gene by knockout, and again you insert homologous right pairs of genes, so that this will again recombine and bind up with that. Okay, so the function is restored. So non-homologous end joining is nothing but a knockout mutation. So this can be anything. So insertions and deletions may occur. That is the why it is called as indel mutations. Indel stands for insertions or deletions. Anything may occur here. So if case in both the case knockout is that, knockout means that the unwanted protein production is stopped. Okay, the gene function is terminated. The protein is not formed. Okay, so this is very useful in diseased conditions. So yes, and here are the pros and cons which I have listed out: high specificity, versatile, and all those things. So, the off-targeticity, that guide RNA should be designed properly, so that it could be made proper. Yes, so that correct you must we must ensure that the correct target site is being bound to the guide RNA, and all those things are there. So and also what priorly ethical issues has to be addressed. See, with this I have included a case study of a CRISPR baby that is being made resistant to HIV. So by a scientist called He Jiankui. So using CRISPR technology, he made that is he made two babies, Lulu and Nana. So they both are resistant to CRISPR, but he hasn't got proper hit and trials. He hasn't what follow the protocols properly for permissions and all those things. So this is again a concern. Okay, so we not as easy as that. We will just get permissions and all those things to mm, no, all permissions and all those things to what perform research on human individuals, and particularly in vivo models. So we have to address that also. Okay, so next I am about to give a brief overview of the whole process with this picture. Okay, so this is the now I hope you people would have got a proper idea so how on this system works, CRISPR Cas system works. So here I am going to that is yeah, I am going to give a brief review. Okay, this is the target site, and this is a non-target complementary site, and again here the guide RNA, some part of a guide RNA is complementary to the target site, and it is bound to that with CRISPR Cas9. Okay, so and again this non-target site, the next to the target site there is something called PAM sequence. PAM sequence what ensures that it makes a double standard break. Okay, and yeah, all those is thing, and this is repair mechanism. Again we can say as it is of two types: non-homologous enjoining and homology directed repair. So it can either be used to for insertions or deletions. Okay, so yeah, finally you got the modified DNA, that is the edited DNA using gene editing technique of CRISPR. So I hope you would have enjoyed this video. So if at all, if I have to make some more videos on some other specific biological topic, then let me know through the comment section, and uh, please give your feedbacks to that. I think that would be helpful and on how to proceed further as a biologist. Okay, so yes, see you soon. Bye. Take care and stay safe.